We're going to get started. It's great to see we've got almost 50 people here. And so this session is going to be split into sort of 3 sessions. Heidi and I are going to tag team for the first talk. And our talk is really to give people an overview, particularly for those of you who are new. And then we will have a separate talk with regard to our GenomeConnect project from Juliann, and then Melissa Landrum from NCBI will talk about Clinvar. So I will get started and share my screen. And so I'm giving sort of the most general of general overviews here. And then obviously, we know many of you are interested in our expert panels. In the second half of this first half hour, Heidi will take over and talk specifically about variant and gene curation. So as you, of course, probably know by now today, the purpose of ClinGen was to create an authoritative central resource that defines the clinical relevance of genes and variants for use in precision medicine and research. And we've updated this graphic actually earlier this spring to try to really highlight, and you'll see it again in Heidi's part, the 4 critical questions we try to answer with regard to the evidence that a gene causes disease. What is the evidence for a given variant in that disease? What's the evidence about an increase or decrease in the number of gene copies? And then I'll reflect briefly on our sort of innovative approach to saying, well, if a patient has a variant in that disease, what's the evidence that it's actionable? I just wanted to highlight that the 3rd phase of funding is going to start in August, and we will have 5 more years of funding, which we're very excited about. We're pending the final notice of award, so just to let you know that we've got sort of really enthusiasm among the team from the 3 different grants that were submitted to continue the great work you're hearing about today over the course of the next 5 years. So as you heard in my first introductory slide, but in case you missed it earlier this morning, or whatever time it is where you live, ClinGen is truly an international network, and I think you heard that beautifully described in the prior session, where we had some of our volunteers from both the United States, Canada, Vietnam, and many other countries participating. And we really encourage international participation, and actually got some nice ideas from the last panel, excuse me, about how to increase that further. This slide sort of highlights the progress. And as I mentioned, we're just about ready. This blue thing is a little early. We're really going to enter the 3rd phase right here this summer. But just to remind you of a few highlights that we began in 2013. And early on, we created a sequence variant interpretation committee to really help assess and provide guidance on the use of the American College of Medical Genomics guidelines and AMP guidelines for variant classification. You'll hear about that from Heidi. NIH supported some genome panels to help support our early expert panels. We developed our gene curation framework. Our interfaces were developed. You saw great evidence of our community curation in the last session. You'll hear from Heidi, and I have one slide about our FDA recognition of variant curation, which is an important piece of the VSEP process. Melissa will talk about Clinvar, and over 1 million Clinvar submissions occurred about a year ago. And then just in the last few weeks, other NIH partner institutes have funded some new genomic panels. So you will hear about new panels developing and some existing ones being supported with funding from some of the other institutes in the National Institutes of Health. And so really one of the goals of this retreat is sort of to get everybody ready for this sort of exponential growth that we hope to see really starting this summer and moving through our next 5 years of funding. Deb Ritter, one of our staff scientists working with our wonderful website team, put together this slide, just highlight we have many different types of curation. So I mentioned dosage, there's variant, there's gene. We'll talk briefly about actionability. This morning, there was a panel about somatic cancer. But all of those curation activities really rely on a whole set of committees that have developed the evidence-based frameworks that we're very particularly proud of, that say, okay, if we're going to curate gene disease evidence, how can we do that in a very structured way? And I was actually delighted to hear some of the members of our expert panels comment on the importance of the evidence for curation. That is something we feel is a fundamental aspect of ClinGen. You heard from some of the groups making the platforms. We have a rich underlying software structure with large teams of software developers who build the tools that we all rely so heavily on. And then you'll hear from Heidi about our expert curation domains. And probably today's retreat is the best evidence of the great work of our education engagement and outreach group, who really worked to kind of spread the ClinGen message as broadly as we can. So of course, many individuals participate in ClinGen expert panels. And again, I won't go through this because Heidi's going to talk about it in detail. But it's just important, as you've already heard, that all of the panels bring together a wide variety of people. So researchers, clinicians, diagnostic labs, scientists to share data. As I mentioned, we have these different kinds of panels. We also have dosage expert panels, although we're not going to talk as much about them during this session. And then actionability is a little bit different because they're large panels for adult and pediatric medicine. They don't split out into individual disease types, and they curate the evidence for a given gene across all adult or pediatric applications. So to just mention this, our actionability framework was published in this article in Genetics and Medicine now several years ago. And the lead investigators in that early effort in terms of the senior leadership were Mark Williams, Jonathan Berg, James Evans, and Katrina Goddard. And now Adam Buchanan now is a co-lead of this effort as well, and Bradford Powell has also taken a leading role. Of course, all of the information about actionability is on our website, and we list all of the pediatric and adult reports by topic, and we have outcome intervention scores and the results. And we're now beginning to do summary scoring. That's just getting underway, so I won't talk about that so much today, but it's just to say I took this right from our website. You can browse any of the curations for the actionability reports by topic. You can look in pediatric or adult, and and also there's just a small document that explains the actual scoring matrix. So this is just an example I pulled of Rett. I'm a cancer person, I always pick cancer examples. And so this is the adult curation on the top. And basically what you can see is they are starting to pull it out by, this is one particular complication of Rett, which is particularly relevant in adults, which is hyperparathyroidism, which is why they use this example. And then they score it for the severity of the disease, how likely is a mutation in this disease to cause it, how disruptive is the nature of the intervention, and then how effective it is. And they're now coming up with a combined score that will try to reflect all of those numbers together. On the pediatric side, it's the same. I'm sorry, I didn't align them perfectly, but it's the same gene, but now the focus is on medullary thyroid carcinoma because the prophylactic surgery is done in childhood. And so they assess that evidence in the pediatric actionability. So to just wrap up before I turn it over to Heidi, you heard a lot about ClinGen Community Curation Working Group. Please, if you're new to ClinGen, and that's why you're in this session, please do the survey that's on our website. Again, it is an international effort. We've trained over, and this slide is old, we trained over 300 individuals, and I think it's many more now because we've already had the June training. And many of the people who volunteer are interested in comprehensive training and then to be assigned to a panel. But you can also do the baseline and curate the literature and provide evidence for us, even if you don't want to do the complete curation. So this is just a lead into Heidi, which she'll talk in a minute. I just have one other slide about the fact that we are very proud of the fact that our variant pathogenicity process was recognized by the FDA. That's the Federal Food and Drug Administration in the United States. And for that effort, it really generated a very strong, even stronger than we already had, feeling of being able to provide all of our curations and the evidence we use on our website or on our tools like the evidence repository in order to make it clear and as useful as possible to the larger community. So to just finish up, as I think you probably gathered from my talk and from others, all of our guidance and curated results can really be found on clinicalgenome.org website. We were thrilled. I remember the meeting when we were looking for URLs early in ClinGen, and we're thrilled to discover that clinicalgenome.org had not been taken by anyone else. And so really you can browse all of the curations. You can find out about the working groups, the expert panels, obviously today's event there, and you'll hear more about the website, I think, tomorrow from Aaron. And then again, for any, once you go on the website for every gene, you can find out exactly what curations have been done. And I always just want to plug in for this particular icon, because underneath it, it shows you all the other kinds of data that you can have shown on the website. And then you can download everything. You know, if you get your perfect table together of what you're interested in, you can automatically download. And with that, I'm going to turn it over to Heidi. I think, Heidi, you're going to share from your computer, correct? Yes.
Yep, that sounds fine.
To talk
Sharon.
more specifically about expert panels.
Yep. So I think what we planned was we'd try to keep it to 20 minutes. So we had 10 minutes or at least 5 minutes time for questions, and then we'll move on to the next topic that Jules will talk about, GenomeConnect. So I'm going to share my screen. And also, if there's, since we will have some time for questions, if there are questions, put them in the chat as you think of them, and then we'll go there first and then open it up for verbal questions after that. Share and present. Oops, sorry. Went on the wrong screen. Stop sharing. Usually it goes to my screen 2, not screen 3. Try this again. All right, can you see the?
Yeah, it looks great. And I can also answer some questions as they come in to save us some time. I mean, on the chat.
Oops. So, sorry, it's advancing it. Mind of its own. All right, so as Sharon mentioned, she talked a bit about the expert panels that we have formed. So this is just a screenshot of the website that lists all the expert panels, and they are divided, you know, across the gene curation ones and the variant curation, but they all sit under these clinical domain working groups that cover major areas: cardiovascular disease, hearing loss, neuromuscular, et cetera. And then those working groups then spawn individual expert panels, and some may have just one right now, and others may have a whole series of them. So last I counted, there are 78 expert panels that we have spanning all of these efforts. So you've heard from the different breakout sessions and talks that, you know, gotten a sense, but just to drive the point home, we have our expert panels are focusing on either gene curation or variant curation. Some actually handle both from the same group of people. But it is important to just step back and say, before you can evaluate a variant and its pathogenicity, you have to know if the gene is actually validly implicated in disease. And that's one of the reasons we spent a lot of time on gene curation. We published the framework for doing the gene disease validity curation, American Journal of Human Genetics a few years ago, shown here. And the details of that are really a framework to curate genetic evidence, so case observations, segregation in family members, case control studies, as well as experimental evidence, gene expression, model organisms, rescue studies, things like that. And we have point values that are assigned, and that's how all of this work gets done in a semi-quantitative way. And then adding up the point values gets you to one of these levels here: definitive, strong, moderate, limited, animal model only, no known disease relationship, disputed or refuted. Now, there's also recently a framework that's been published by the American College of Medical Genetics Laboratory QA Committee, guiding how to use these categories in designing gene panels for clinical use. And what you'll see here, just at a high level, is recommendation that use of definitive, strong, and moderate are appropriate to put into diagnostic targeted panels, whereas you should leave the genes with less certain evidence, like limited and below in this sort of reddish categories, for exome and genome for the most part. And that's the basic guidance. And you can read more details in that article from ACMG. On our website that Sharon was showing you a few screenshots of, one thing that's there are the stats on all of the gene curations, both overall across all these groups, as well as a little circle for every group. So I'm just showing you a glimpse of some of them here. Overall, from the last time I captured this, there were 1,357 gene disease curations that had been done by a ClinGen expert panel. I do highlight here one important point: about 29% of the gene disease relationships curated are what we consider insufficient evidence. And from some of the work of the expert panels, it's clear that most of these never move up. A small subset, particularly some recent limited ones, can move up, but most don't. And I think that's just a really important point, because, and if you look across all of these areas, they all suffer from reports of gene disease relationships that really don't have sufficient evidence, and keep that in mind. I also took a screenshot of one gene here just to point out that we often curate genes for more than one disease. You can see for MYBPC3, it's been curated for hypertrophic cardiomyopathy, but also arrhythmogenic right ventricular cardiomyopathy and dilated cardiomyopathy. And the levels of evidence are different. It's definitive for HCM, but limited for DCM and ARVC. And that happens quite frequently. One thing I do also want to point out, ClinGen is collaborating in part of a larger effort that we're working on called GenCC, the Gene Curation Coalition. So all of ClinGen's data flows into this system, but so do all of these groups shown here on the right. Got a whole lot of logos. You'll recognize Marina because she's been in lots of the sessions. She has been helping lead this effort. Collaboration, and Scott developed the database that we just recently launched. But all of the gene curation work from both a variety of public resources and companies that are doing this within their own laboratories are all sharing this evidence. You can go and see gene curations from multiple groups, not just ClinGen, on this GenCC website. We sometimes call it a Clinvar for genes. So I just showed a screenshot of one gene here, but there's lots of data coming in, and we're working on getting more and more data in this system. Now, if we switch over to variant pathogenicity, and I should mention, there's a finite number of genes in our genome, and we do, as ClinGen, hope to curate them all and all of the gene disease relationships that have been claimed. I think that's an achievable target. On the other hand, variant pathogenicity is a whole nother beast. There's over 800 million variants that have been identified in our genomes, and many more to come. So we, as experts, will never curate all of those variants. So we have to think about other ways to tackle this challenge and ways to prioritize and disseminate our approaches when we get to variants. So how do we do that? Really through 4 different approaches. The first is using common standards and terminologies and rules for variant classification. Stephen and Marina and others gave a sort of breakout session on specifying these rules for different gene and disease areas across our expert panels, but all of the labs are using this framework in order to apply it to variants they curate either in their own laboratory or throughout the world. So there's a lot of work in developing both the standards at a gene and disease specific level, as well as this work group constantly evolving the rules. And we have reconvened the ACMG AMP committee, now partnered with ClinGen, to come out with the next set of rules. So as we build these rules and disseminate them, it helps our whole community do curation at a more rigorous level. And all of the additional recommendations that are either published or put out as white papers from the SVI working group, and they can be found on the ClinGen website shown here. Next is working with our community to share their variant classification work. That allows us to create transparency in this work, as well as crowdsource it by all of us sharing our work in a common place. And of course, we've partnered with ClinGen, we've partnered with Clinvar at NCBI, who built this database and has been collecting submissions from thousands of submitters around the world. Most of the data is from clinical testing. And in fact, 84% of the data in Clinvar is from the top 20 laboratories. So, and these are mostly commercial clinical labs. So it really shows how much our community has come together in this space and been willing to share this data voluntarily for the improvement of patient genetic testing interpretation. So it's just been a really great experience. We also allow patients to directly share their data through the GenomeConnect patient registry that ClinGen has. Much of this work is led by Juliann Savatt, who you'll hear about next. So I won't steal her thunder. And then next, we work on interlaboratory conflict resolution. We don't yet have an expert panel for every gene and disease area. However, when conflicts arise, it's useful for labs to come together and share their evidence and resolve those differences. And in fact, if you look in Clinvar, you will find that there's about 1% of the variants that have what we consider medically significant conflicts. So one lab calling it path or likely path, another lab calling it uncertain, likely benign, or benign. Those lead to difference in how patients are managed. So those are the ones that we prioritize a focus on, both between labs as well as in our expert panels. And Stephen, who you heard from in one of the breakout sessions, and we'll hear more from tomorrow, led some effort along with Jill Jelinski at Ambry in discrepancy resolution between laboratories and really showing the benefits of it. I don't have time to talk about that in detail, but it's an important point. Lastly, we are engaging experts, and you've certainly heard in the various sessions about the expert panels and the work that they do, defining their rules and piloting those rules and then implementing them. All of the framework is recognized by the FDA so that our variant classifications go in with that tag into Clinvar. And those groups both resolve differences in interpretation, but also over on the right here are showing VUSs that have been able to be classified because labs have come together and shared their underlying evidence. And so that's also a really important activity of our expert panels. You've heard a little bit about coming together as a community and volunteering and participating. That information, how to do that, is on our website. You can sign up. And with that, I'll just acknowledge and thank many of the leadership of ClinGen and the working groups that develop all our methods and approaches and tools shown here, and a big happy thanks from the whole team. With that, I'm going to stop, and we have, I think, at least 5 minutes for questions before we have to move on to the next session. I see there's some stuff in the chat.
There was a question about some of the surveys in communities. So I answered that, but there must be other people. There are 71 people out here. Are there others who have questions or questions about how to get involved?
You can either pop off of your microphone and ask out loud or type it into the chat.
So Heidi, you and I talk a lot about one thing I think that in your time you didn't get a chance to say about which variants we do actually prioritize VSEPs to curate. So, you and I have talked in the cancer space, there's a lot of interest in curating the secondary finding genes because many of those have prophylactic surgery recommendations. And so people want an expert panel opinion. So we've been fairly deep in our approaches, or hope to be deep. But
yeah, it's a good point, Sharon. And
I'm sorry, my camera stopped working.
Yeah, in addition to, prioritizing variants that are discrepant or VUSs from multiple labs, Sharon's pointing out just gene and disease areas that we prioritize. Certainly when there are clinical trials for new therapeutics, those are really ripe for launching expert panels because a lot of the criteria for patients to enroll in those clinical trials is having pathogenic variants that are clearly, accurately interpreted. And the outcomes of those trials are going to be best if those variants are accurately interpreted. So whether it's forming an expert panel because of, priorities around secondary findings or clinical trials, other things like that, those are reasons to prioritize and to even allow patients to say, into this clinical trial, here are my variants, please curate them for me. That's something that an expert panel might do as well, to respond to the needs of the community and therapeutic development.
Yeah, and I just chose a different camera. And I would just say the other one, which we've talked about, for rare diseases where there are a smaller number of pathogenic variants, the expert panel can still be very useful in creating the guidelines so that, and we publish them all and they're all on our website, so that if a diagnostic lab now finds a new variant in that gene, those guidelines are designed to help you curate the variant in your own laboratory, as well as the variants that we classify ourselves. And
Absolutely.
with that, maybe we should just give Juliann a couple of.
Well, there's one more
yes.
question that just came in, so I'll just quickly answer that. So a question came in. I'm curious whether there is a way to learn about upcoming curation plans on variants, genes, and diseases. So 2 comments here. One, if you go to the website where the expert panels are listed, many are listed right after they've been formed, particularly the variant ones, before they've actually gotten through all their steps. So you can see that a group has formed and what their scope is from that initial application. So that's one way to see what's coming. Now, sometimes it just takes, a month or so to just, organize people and make the list and gather the conflict of interest. So if anybody has a question of whether we're going to be launching something, just reach out, email clinicalgenome.org, the general email on the website, and say, hey, has anyone thought about launching a panel in this area? Certainly, if you're interested in starting one, we'd love to hear from you, but you can also just ask if anyone else is. So we try to get the website up to date as quickly as possible, as soon as these groups are formed, even before they start their work. But if not, then feel free to email us and ask if something's in the works.
And internally, we do have a tracking system, which eventually will be more obvious to the public. So we are all aware of which genes are up next and which expert panels are being formed.
And we take suggestions too. If you think there's a reason that a gene or should be prioritized, or a disease area, and want to share that with us, please, send it our way. And Erin put the email in the chat there, ClinGen@clinicalgenome.org. All right, okay. I think it is, we have 35 more seconds according to my timer, so I think it's time to turn it over to Jules.
Thanks, Heidi and Sharon. Let me go ahead and share my screen. All right. Hopefully it's in presenter mode for all of you now. So as Heidi and Sharon mentioned, my name is Juliann Savatt. I'm a genetic counselor at Geisinger, where I help to coordinate ClinGen's patient data sharing efforts, which includes GenomeConnect, our patient registry, as well as some work with other gene and disease specific registries. So I'm really delighted to speak to you about the ways that we're engaging patients in data sharing. So today, to give you a sense of what I'll talk about, I first will talk a little bit about how patients can participate in the data sharing process by submitting their genetic and health information to patient registries, including GenomeConnect. We'll discuss how patient-shared data can benefit both the genomics community as well as individual participants. And finally, I'll end by outlining how you can get involved by either using patient-provided information or encouraging such data sharing. So similar to Heidi and Sharon, I think I'll take about 20 minutes for my talk, so we should have plenty of time for questions. So feel free to drop those in the chat as you think of them as I'm talking, and then we'll address those at the end here. Before I go ahead and launch into my slides and telling you about GenomeConnect and patient data sharing, I thought it might be helpful for me to know just a little bit about who's all here and your involvement with ClinGen. So I'm going to go ahead and drop a link into the chat, but you also can use the QR code to answer that the link. Let me put this here. So the link is in the chat. Let me put it back on the screen. And I'll move over. Thanks for the audience participation. I know it's late in the day on Thursday after listening to lots of talks. I'll give it just a couple of moments. Well, it looks like, I'll let these keep coming in, but we have sort of a diverse group here. Some involved in VSEPs, others GSEPs, others just here to listen and learn. So hopefully there'll be some takeaways from today. And I saw there's not an option for I don't know. That's okay. If you don't know, I think we'll be able to address pieces of how patient data sharing can potentially impact your work that you're doing or that you might do in the future. So you've seen this graphic quite a few times today already, but I think it's useful just to explain what we're going to talk about today, how it fits into the broader ClinGen effort. So I think Heidi and Sharon talked a lot about ClinGen's critical questions, and Heidi alluded to the fact that those efforts really rely on crowdsourcing, on data sharing. She talked about how ClinGen is working with Clinvar to encourage data sharing from labs, clinicians, and researchers. And as she mentioned briefly as well, we also encourage patients to share data. And that's really what we're focusing in on here. Labs, researchers, and clinicians have been sharing genetic and health information for some time, and Heidi put some numbers around how many variants have been shared. But ClinGen felt strongly that patients could also serve as an important source of additional information. Patients are often experts about their own health history. They can provide much more details about their symptoms over time, their diagnoses, and their health. And ClinGen felt that access to that additional information could be helpful to increase our understanding of genes and genetic variants. As a result, back in 2014, the ClinGen team launched a patient registry called GenomeConnect, really to empower patients to share their de-identified genetic and health information with publicly available databases like Clinvar. GenomeConnect is open to anyone who's had genetic testing regardless of results or diagnosis, so it's not gene or condition specific. Participants enroll online, consent to de-identified data sharing and recontact from the registry. They provide their health history through patient surveys, and they upload a copy of their genetic test results to their online account. As I mentioned, we collect health history via surveys. So after consent, every participant receives an initial broad health survey. This survey inquires about general issues across 17 sort of broad categories or body systems. So things like issues with the cardiovascular system, issues with the endocrine system, GI issues. Each question provides examples of issues in that system. And then for those that individuals indicate they've had an issue with in the past or currently have an issue with, they then are assigned additional questions, trying to drill down further, what are their particular symptoms and diagnoses. We also assign additional surveys over time to collect even more information about their health history. So for example, an individual on one of the first pages of the survey would see a question about, have you had issues with your heart and or blood vessels? A simple yes, no, unsure. If they say yes, they receive an additional page that lists out cardiovascular issues, arrhythmias, heart defects, cardiomyopathy, which you see here on the screen. Each individual phenotype is written out in patient-friendly language, but on the back end, if they indicate, yes, I have a history of cardiomyopathy, each of these symptoms is mapped to a human phenotype or HPO ontology term, which allows structured data sharing outside of GenomeConnect. In addition to this initial survey, individuals who say, yes, I have a cardiomyopathy, would then receive a second survey later on, asking more questions about the type of cardiomyopathy, any additional risk factors that are non-genetic, like a history of chemotherapy, again, to try and collect more phenotype information, more information about their health. Participants share their genetic test results by uploading either a PDF or image of their result to their account, or by providing our team with permission to request their records from the lab that performed their testing or their healthcare provider that ordered it. Genetic counselors on our team then review those reports to ensure that all of the genomic data are collected in a structured and standardized way, and really to ensure its accuracy so that we can then go share that information more broadly. Once genomic and health information are collected from patients, their de-identified variant and health data are shared with Clinvar, which Heidi mentioned, and you'll hear much more about from Melissa shortly. But as you probably know, it's a publicly available database of variants and their classifications. And as you also know, this is where labs, clinicians, and as Heidi reviewed, often submit variants. So submitting information here from a patient registry makes it publicly accessible to those other stakeholders so that they're aware that this variant has been identified and potentially can see more detailed health information about the individual that was found to have that variant. The data are submitted to Clinvar using a phenotyping only submission type, which highlights that this information is coming from someone who's not reinterpreting the variant, didn't identify the variant through clinical testing, but is submitting it as it was reported by the clinical laboratory, but along with additional phenotype information. This submission type isn't counted towards star status or aggregate classification, which prevents a duplicate counting of a single patient. And I'll show you later on some slides, but we do all we can to try to highlight reporting institutions classification, so a Clinvar user can readily see this lab submitted, and also that participant must have gone and enrolled in GenomeConnect. In addition to submitting to Clinvar, we submit some genes to Matchmaker Exchange. A subset of participants share reports that identify candidate genes. And as you might know, candidate genes are those that may be associated with a set of symptoms based on some preliminary research, but really much more information is needed to better understand if that particular gene is associated with a Mendelian disease. Candidate genes that we identify on participant reports are submitted to Matchmaker Exchange, which is a data network that's really working to enable matching with other healthcare providers and labs who are exploring gene-disease relationships. Our hope is that by sharing information here, we can contribute to moving these candidate genes into a more definitive category of having a disease association or not being associated with Mendelian disease. So why is patient data sharing important? So I certainly have many a slide to talk through our experience to date, but I thought it might be helpful at this point, since I'm halfway through, for some other participation from you all. So that same link that's in the chat, this is a word cloud. You can go ahead and type in if there are any initial thoughts you have about how could this information be useful. And if not, don't worry, I'm going to tell you in the next few slides here. Give it another second. There we go. Some are coming in. Support, connection, improved precision medicine, genome studies, genetic mastery, discovery. So I think you've hit it with these, you've hit the nail on the head, that benefits are sort of twofold. And as I alluded to, I think earlier in our discussion, that patient data sharing can be useful to the genomics community, lead to discovery. Also, there are some benefits back to individual participants, like connections and support. So I want to talk through those a little bit further in these next few slides. So first, how can patient-provided data inform our genomic understanding, our understanding of variants and gene disease classifications? So I think Heidi summed this up well, but when a genetic variant is identified, the critical questions really are: first, is that gene associated with disease? That's our gene curation efforts are focused on that. And then second, if so, is that variant pathogenic? Is it going to impact that gene's function in the body? Patients participating in GenomeConnect, what we've seen over the past nearly 7 years is first that they're providing additional phenotypic information. This was really the goal of GenomeConnect from the outset, that patients could provide much more details than are often included in lab or other submissions. We're seeing that in practice. They're providing health history data, they're providing information regarding inheritance of the variant, and we also have the ability to recontact them over time if additional information is needed or could be helpful to inform our understanding. Really knowing patient-specific symptoms, knowing the inheritance of a variant, knowing if other variants were identified through testing, all of that can help shift variants of uncertain significance into more definitive categories, or could inform a gene-disease relationship, perhaps. In addition to providing additional details like enhanced health information and information regarding inheritance, Patients enrolling in GenomeConnect are also providing novel genetic changes that haven't been submitted to Clinvar previously. From our submissions to date, 41% of variants weren't submitted by the reporting laboratory or another institution previously. So really that participant taking the time to enroll in the registry is what got it out there into Clinvar. Putting this information out there in the public space can be useful the next time that particular variant is identified or if a similar variant is identified in future patients. Altogether, patient data sharing can provide additional data that can inform both genes and variants. So if we're thinking about those gene curation and variant curation efforts, patient data sharing can provide access to unpublished cases that could be used to inform gene disease relationships. Patients also can provide case level details that can be used to inform variant classifications. To help illustrate the power of patient data sharing, I wanted to share a brief case example that is actually something that occurred and that we can appreciate from our data sharing. So for the purpose of this case example, I will call this participant Ella. She's a pediatric patient with a history of seizures beginning in infancy, and she had exome sequencing due to her history back in 2015. A likely pathogenic variant in SCN8A, a gene associated with the seizure condition, was found. Her parents were tested as part of her trio exome, and the variant was found to be de novo. Because the variant had not been seen in healthy individuals, was found to be de novo, was expected to impact the protein, the clinical lab classified it as likely pathogenic. Ella and her family had an answer for her history, but they chose to participate in GenomeConnect to share her information more broadly. We then submitted her de-identified genetic and health information to Clinvar. At the time of our submission, there were 2 other records in the database. So lab 1, the lab that had actually reported the variant out to this family, had also submitted to Clinvar, though their record contained fewer details about her clinical history and didn't have information about inheritance of the variant in her family. Labs often have these details, but they might not be able to share them with Clinvar. We also saw a submission from Lab 2, who had classified the variant a year prior as a variant of uncertain significance. After our submission, Lab 2 reached out to the registry team after seeing our Clinvar record and wanted to know more about Ella's seizure history and to confirm that the variant was in fact de novo. In their patient, parental samples weren't available, so they weren't able to complete parental testing. And I think, as you all know, understanding if a variant is de novo can be incredibly helpful in informing that variant classification. So we were able to confirm by re-reviewing Ella's report that the variant was in fact de novo, which prompted Lab 2 to reclassify the variant as likely pathogenic and update the family that they had identified that variant in. Ella's results in care weren't impacted by her family taking the time to share her results, but ultimately them sharing enabled another family to get clear genetic results. So in addition to informing variant classification and gene disease validity, patient-centered data sharing can also benefit the individual patients themselves. So first, it can facilitate genetic updates. So as I just outlined in that case example, our understanding of variants can shift over time. In fact, we hope to reclassify variants of uncertain significance, and rarely we also know that our understanding of pathogenic and benign variants might shift as well. In just one study of a cancer genetics lab, over a 10-year period, 6.4% of variants were reclassified, leading to more than 60,000 updated reports. So we know this happens in practice. Clinical lab and healthcare providers' duty to reassess genomic results and recontact patients has been the subject of ongoing discussion, and there's no clear legal duty to recontact, but there is a consensus that recontact is beneficial, and most patients seem to value being recontacted, hearing about updates to their results. Lab and healthcare providers' reassessment and recontacting practices really vary, and there are certainly barriers to recontact, including limited resources, limited reimbursement, changing contact information of patients. So under current circumstances, 2 large genetic societies, both ACMG and ESHG, have suggested that patients share in the responsibility of recontact. ACMG has also suggested that novel methods of recontact are needed to ensure that patients are remaining informed of their results. So early on in GenomeConnect's data sharing, we recognized that the registry might identify cases where the participant's report is out of date compared to the current knowledge base. whether that's in Clinvar, we can see that the reporting lab has updated their classification, or via our submissions to Matchmaker Exchange, we might come to appreciate that a candidate gene is now disease-causing. As a result, we reached out to our participants with a survey back in 2016 to just learn a little bit about would they want to receive updates from the registry? What are their thoughts and reactions to that? After an overwhelmingly positive response that participants wanted to receive updates in general and wanted to receive information from GenomeConnect, we designed a process really based on their feedback. That in February of 2017, we prompted all prior participants to provide their preference regarding the receipt of updates. And now upon enrollment, anyone who's newly enrolled is required to tell us if they'd like to receive an update from us, if we should identify one from our data sharing. Of course, we won't identify all updates, and that's all in our consent language, but we like to give the choice of if they would like to receive this information, that we will pass it along should we learn of anything. To date, 98% of those who've responded and said, provided a preference have opted to receive updates from the registry. Should we identify an update to results, we first reach out to the reporting laboratory if needed to confirm the variant classification in Clinvar or that gene disease relationship. And then from there, reach out to the participant, informing them that there might be an update to their result and encouraging them to reach out to their physician or genetic counselor or other provider to learn more about that particular update and its impact on their care or their results. In total, GenomeConnect has provided 45 updates back to participants to date. They're outlined here on this slide. 17 such updates that fall into that medically significant or potentially medically significant bucket that Heidi had alluded to. So it might impact diagnosis and/or management. So ultimately, patient data sharing can enable individuals and families to remain informed about their genetic test results. Finally, patient data sharing can support connections between stakeholders. So I think as some of you in that word cloud thought of, registries can connect others. So first, GenomeConnect enables connections with researchers or clinicians who are interested in a particular gene or variant. If a researcher or clinician reaches out to us, we're able to recontact participants to collect additional information or to inform them of additional research opportunities. And additionally, participants can connect with one another based on gene, disease, or US state through the registry. Many genetic conditions are rare, and patients might have the experience of being told that they're the first patient their healthcare provider has seen with that diagnosis or a variant in that gene. And the registry can really enable them to connect with others to exchange both informational and emotional support. Given GenomeConnect's experience with patient data sharing, I think we really recognize that patient registries could serve as an important source of additional information while also benefiting the individual participants. We also recognized that GenomeConnect is just one of many patient registries. There are other gene and condition-specific registries out there that are potentially collecting really valuable information, but might not be sharing it in a public way. As a result, in 2018, we started working with registries who were engaging patients with genetic testing or genetic diagnoses to really use the process that we developed to share data with Clinvar and help them do the same within their registry. We work with registries that are on the same registry platform as GenomeConnect through Invitae, as well as registries on other platforms. And to date, we've engaged with 10 registries to help them submit to Clinvar as well. Altogether, GenomeConnect and this work with other registries have allowed us to engage nearly 5,000 patients in data sharing, and more than 1,900 variants have been submitted to Clinvar. And I will say we have a large submission underway, so we expect that number to tick up soon. So finally, how can you get involved? And regardless of how you're involved in ClinGen. So first, you can use the information shared by patients to inform the understanding of the relationship between genes, variants, and health. The variant and health details that we collect from participants are periodically shared with Clinvar, and I'll walk through a Clinvar record here briefly just to show you what it might look like. So use that if you're involved in ClinGen efforts or if you're involved in caring for patients, that's something you can use. We're also exploring a process to export data for relevant VSEPs and GSEPs. So if you're involved in a VSEP or GSEP, we might have been in touch with you already to highlight potentially genes that have been curated as having limited, refuted, or disputed evidence that we have participant cases around. Or if there's a VSEP where we have participants with variants in that gene, we might have been in touch with you already or told you a little bit about what we have. So just briefly, a Clinvar record here. So if you search a variant and come to the variant page, if you scroll down, you might see a record from GenomeConnect. So as a phenotyping only submission, we're not interpreting the variant ourselves. So we do not provide a classification, but we do provide information about how the reporting laboratory has classified that variant. On the main page here, that information can be found in the comment. So you can see that it was interpreted as likely pathogenic the day it was reported, and then the GTR ID, the institution that reported this out. Inheritance information, if we have it, also can be found on this main page. And then much more detail can be found if you go to the supporting information. And then if you choose your columns, you can see all the information that we've submitted from participants, including their HPO terms and age range, sex, so all on this page. We also welcome questions. So it's not uncommon for us to get questions from those who see a variant record, and we're happy to answer that information. To find all of the submissions from GenomeConnect in the registries that we work with, you can go to the submitter list on the Clinvar website, and anyone that we've helped facilitate a Clinvar submission from will have GenomeConnect in the name. So our submission is GenomeConnect. If it's another registry, it'll have their name and then ours as well. Additionally, you can work with us to expand patient data sharing. So if you work with patients, inform them of opportunities to share data through GenomeConnect. We have a number of resources on the ClinGen website to help you do so, and many clinical laboratories include wording about the registry directly on reports. If you work with a patient registry, discuss the opportunity to engage with us in what we call our patient data sharing program, so how we help them submit data to Clinvar. So in summary, I hope today in my time that you all have come to understand that GenomeConnect and other patient registries offer a structured way for patients to share data. And that information can be useful because it provides additional details as well as novel genomic information that can inform our gene and variant curation efforts. Also, registries can serve as a liaison between labs, researchers, and patients to ensure that patients are remaining informed of their genetic test results, and also that we can pass on information about additional research opportunities. Finally, ClinGen is actively working to continue to expand patient data sharing efforts by working with more patients and other registry groups. So with that, I saw the chat was active while I was talking, so I will acknowledge those who make this work possible, including participants and the team at Geisinger, as well as the Broad. Our general email is up here on the slide, and why don't I stop sharing so I can look at the chat and hopefully answer the questions that you guys might have.
The good news is that Melissa was active in answering some of those questions, one of which relates to her own talk. So and one was just defining a de novo variant, which Melissa answered, and then about submission formats, which I think Melissa will get to shortly. If there are any other questions for Jules, we still have a few minutes.
I have
Jules,
a quick question. Oh, sorry.
Oh, go ahead. You go ahead.
Hi, I'm Jane with one of the renal fellow at MGH. So if in case I want to help the patient submit the report, do I do it with them, or I can ask their permission and then I just send the report myself?
That's a great question. So at this time, it's really patient initiated. So they'll need to go on the website and consent to data sharing, and there'll be a consent form that they need to complete. So we don't want providers enrolling them for them, but if you just tell them information about the registry and ensure they have a copy of their report, I think those are the two main things that I encourage clinicians to do. So we do have on the website an informational handout, like a little printout that you can give to patients. We have little like postcard size things that we're happy to send to clinics if they want to attach those to family letters or to reports to help inform patients about the registry. Great question.
You know, one thing I'll add to that is, you know, if for some reason a patient just didn't want to register themselves in GenomeConnect, you can, as a physician, facilitate their data sharing because you can submit to Clinvar. And my guess is Melissa will cover this, but you know, there's a submission wizard that's designed for like one variant at a time or very small numbers of variants. And we actually, as ClinGen, have a one-page consent form that is right on our website. There's videos to explain it to patients. It's very simple, and you can just get the patient's consent, and then you could submit that variant to Clinvar on their behalf in that format, either through the submission. You'll see the different ways that Melissa will share with you on how to submit. So there are options for physicians to directly submit to Clinvar for your patients, but as Jules pointed out, to register in GenomeConnect, that has to be the patient themselves.
Jules, you have one other question in the chat from Greg Lennon.
Thanks, Melissa. So Greg asked, if for those of you who can't see the chat, if GenomeConnect can support reporting of compound heterozygotes or genosets, or if it's 100% single variant by itself oriented. And so the way we collect data is sort of by patient. So if they have multiple variants identified, we're collecting all of that together. And I think that we have continued to iterate on our Clinvar submission, and I don't believe that our submissions to date have always made that terribly clear of when individuals are compound heterozygous, but that's something that we have for all of our participants. So if that's a question, if you see a record and have a question about were other variants detected, was this the only variant in this gene, are they compound heterozygous, do you know if it's cis or trans, those are the sorts of questions that our team is happy to answer if it's not immediately apparent within the Clinvar record.
One more question coming in. Okay, I'm a little confused. Sorry if this is an opposite question, but I just want to clarify. So data is organized by patient or by gene/variant?
That is a great question. So within the registry, it's organized by patient. So each patient enrolls, we have all of their data together, their health information, all the variants that were detected. When we submit to Clinvar, it is sort of more variant based. So we're submitting each individual variant to the database. And I think Melissa will talk a little bit more about Clinvar and how that's set up. But so each variant that is found in a participant would have sort of its own record separately within Clinvar.
Yep.
Perfect question, and perhaps a good segue.
Yep. I think we're just out of time, and so we should turn it over to Melissa and let her do the next stretch.
All right. Thank you very much. I'm going to share my screen here. All right. And do you see slides?
Yep. Not yet in presentation mode, but we see that up there we go. Looks great.
Great. All right. Thank you, everybody, for sticking through this session to the end. I'm Melissa Landrum. I lead the Clinvar team at NCBI, and I'm going to talk to you today about submitting to Clinvar. So I'm going to go through kind of three areas here. I'm first going to give you an overview of the submission process. I'm then going to talk conceptually a bit about some of the data that you submit to Clinvar that can help you as you prepare your data for submission. And then we'll go through some actual examples of some submission spreadsheets that illustrate the three different examples that we'll look at when we look at this conceptually. So to start with the overall submission process, you would start by registering your organization in the Clinvar submission portal. So you come to the portal and log in with your NCBI account. If you don't have one yet, it's easy to set up. There's an online form that lets you fill in information about your organization and the people associated with it. This is important because data that is submitted to Clinvar is really attributed to an organization like a laboratory rather than to an individual person. And then you send this information to Clinvar. One of our curators will review it. And once it's approved, you're ready to submit your data, your variant classifications. So as we alluded in the previous questions, we do have several formats for submission of data. I think Heidi mentioned that we have a single submission wizard. This is an online form. This is the one that she mentioned might be useful for you if you're a physician, if you have a single variant that you want to submit. So we do consider this to be good for a small number of variants and those who will submit infrequently, because it's an online form that walks you through the process of submitting a single record, a single variant that you've interpreted. We also take batch submissions in files. So this could be either an Excel using our Excel spreadsheet template. That's what we're going to look at in the examples. You can also submit a tab or a comma-separated file that's based on the format of the spreadsheet. And this kind of batch submission is good when you've got more than a handful of variants to submit, and it's good for repeat submitters. There's a little bit of a learning curve to the spreadsheet, but if you're going to submit repeatedly or you have many variants to submit, it's worth that extra time. More recently, we've also added an API for submission. So this is good for organizations that have large volumes of variants that they're going to submit. And for those who are repeat submitters who have programmatic support, because you do need coders on your end to both call this API and then process the data that you get back from it. For the talk today, I'm not really going to focus on use of the API. I'm more going to focus on, really focus more on the file submissions, but the downstream steps for Wizard and File are very similar. The data concepts are the same no matter how you submit your data to Clinvar, and I'm also happy to take any questions about the API at the end if anyone wants more information about that. So Wizard and File submissions are uploaded in the submission portal. And what you can expect after that is first some data validation. In the Wizard, the data is actually validated right on the page, so you can't proceed until you fix your errors. For file submissions, you have two options when you send your file to us. So one option is that you can ask us to stop the process and return any errors within that batch so that you can fix them and then give us an updated file so that the whole file passes. The other option is that you can choose to give us your file and we will process anything that passes that step of validation and return any errors to you. And you can fix them on your own time and submit them in a new submission later. Repeat submitters often choose this second option because it lets them get their good data out faster and they can just take their time and fix the errors later. So once we get your submission that's passed this first round of validation, one of our curators will review it and process the data to our database. In this process, they sometimes find additional errors or they may just have questions for you to clarify some of your data. So if that happens, they'll email you and work out any issues. And once all of that is sorted out, we will load it to our database. And we publish data weekly, which makes it available on our website, in our FTP files, and by eUtilities, which is our API for retrieval of data. So you can expect to see your data published the Monday after we've loaded it to our database. You also receive a summary of your successful submission, and that includes information like your SCV accession numbers. We assign an accession number to each of your records, and you need that accession number for when you'll update your data. And that is very important in Clinvar. This kind of data, variant classifications, and evidence for it can change over time, and it's very important that if you've reclassified a variant or you've found new evidence, it's important that you update your record in Clinvar so that others know about that change. One thing that is important to note is that within Clinvar, we consider you as the submitter as the owner of your submitted records. So you and your organization are the only ones who can update that data. We will not update it for you. Other Clinvar users cannot correct your record for you. So if there's any update that needs to be made, your organization takes responsibility for that. You can do that update anytime after your records are published, and there's two ways to do it. You can update it in a file submission. We'll take a look at that in one of our examples. That's useful if you submit frequently and do batches of file submissions. You can just add any updates to your next batch. We also have an online form called the Single SCV Update Form. So this is helpful if you've got, say, a single variant that you've reclassified, and maybe it's one of those medically significant changes to the classification, and you want to get that change out there quickly without waiting for your next batch of submissions. So that's the overview of how to submit. We're next going to talk a little bit conceptually about the data that you submit, so that this helps you think about how to map your data to Clinvar and help you as you prepare your data for submission. So every Clinvar record has these 4 domains of data. So there's a variant. There's a condition that you've interpreted the variant for. It's usually a disease, but could also be a drug response. There's your interpretation of the variant. And it's important to note that we consider this to be a general interpretation of the variant for disease or drug response. It's not a patient-specific interpretation. So if you've seen a variant in more than one patient, we are expecting that you are submitting your aggregate interpretation based on all of your observations of the variant. And so that can be shown as you look at the evidence for an interpretation. So this is the fourth area of data, and the evidence in Clinvar is structured as observations of the variant. The minimal fields that we ask for an observation of variant are shown here. So that's the allele origin. That's basically whether the variant was observed in the germline or as a somatic variant. The affected status of individuals with that variant, and the collection method. So this is asking, how did you collect this data and interpret the variant? Is this part of clinical testing or research? Or for example, this is where Jules and her GenomeConnect submissions would indicate that it's phenotyping only. So this structure of providing evidence is fairly flexible, and it can be used in a few different ways depending on the submitter and what kind of information they can submit. So we're going to look at a couple of different examples here conceptually, and then we're going to look at the same examples on a spreadsheet. So first of all, we're going to look at submitting an SNV with some very basic information as a single aggregate observation. This is a good example to look at because this is very typical for submissions we get from the big clinical testing laboratories or from the ClinGen expert panels. So in this type of submission, they will, the submitter will tell us the variant that they've interpreted, the disease or the drug response condition. They will give us their interpretation of the variant, so something like, benign or pathogenic. And then they might give us some information that's related to that interpretation. So they might give us a comment. So that's that blurb that you see on the Clinvar website that explains why they interpreted that variant as they did. They might give us citations like PubMed IDs, the date that they last evaluated the variant, and the mode of inheritance. So in this case, a lot of the information is actually attached to the interpretation, and in their evidence, they might just describe a single aggregate observation, and this might represent seeing the variant in one person or 5. They may not really specify, except in free text in the comment, but they might just generally say, we've observed this variant in the germline. We don't know the affected status of individuals with the variant because it's often the clinical testing labs. They haven't actually seen the patient, but they do know they've collected this data through clinical testing. So this is a very typical kind of submission, as I said, from clinical testing labs and expert panels. And again, this is for an SNV. CNVs often look a little bit different. So often for a CNV, there's a single observation, but because of the nature of CNVs, they don't necessarily aggregate together. So CNVs typically have a single observation, but it's more at the case level. So these records can look a little bit different. Again, the submitter would tell us their variant and the condition. We'll talk a little bit more about condition when we look at the submission spreadsheets. They would give us their interpretation of the variant and maybe a comment and the date it was last evaluated. And in their evidence, they might give us, again, a single observation because they probably only observed this in one patient. Again, with these minimal fields of the origin, they might tell us that it's germline, the affected status, and that's from clinical testing. For CNVs, we also ask for this field called the structural variant method and analysis type that tell us how the CNV was detected. And typically, those who are reporting reporting CNVs also know something about the clinical features that were observed in the patient. So this piece of information is a little bit more common in submissions for CNVs than for, say, SNVs or small indels. So again, this is typical for a clinical testing submission. Research submissions can look quite different because often researchers have more consent and they can provide more specific information per individual in whom the variant was observed. So for the third example, we're going to look at reporting a variant that was observed in a trio where the observations are going to be reported at the case level for each person in that trio. So again, the first 3 pieces of information might look very similar: the variant, the condition, the interpretation, and these related pieces of information. The evidence might be much more detailed. So the evidence might actually have 3 observations. So let's say 1 for the proband, 1 for the mom, 1 for the dad. And for each person, the submitter might be able to give us these minimal fields and then much more information like their age, their sex, their zygosity for this variant, any observed clinical features, their ethnicity and population group, or other pieces of information. So you can see this kind of structure for the data that you submit to Clinvar can vary depending on the information that you have, the consent that you have to share case level data, and also the time and ability that a submitter has to structure data specifically for a Clinvar submission. So based on those 3 examples, we're now going to look at really examples of submission spreadsheets and look and see what those 3 cases look like in the spreadsheet. So the first example we looked at, to remind you, is reporting an SNV that just has a single aggregate observation. And so this is the typical kind of submission that we would get from a clinical lab reporting an SNV or an expert panel. So for these examples, I'm using our full submission spreadsheet. We have a smaller one called the light spreadsheet, but for the purposes of this demonstration, I'm using the full spreadsheet. I've also hidden a couple of rows here that have lots of instructions just to make it easier to look at. But when you go to do a submission on the spreadsheet, I do encourage you to read those instructions. They have lots of useful information. So to get started on our submission, we're going to skip these first 2 columns. We don't need, most submissions don't need these identifiers. And we're going to go right to describing the first piece of information in a submission, which is the variant. So the gene symbol is optional, but that is a useful piece of information to provide. But what we really need is a specific description of the variant, and that can be using HGVS. So if you do that, you would fill in the reference sequence for your HGVS expression with the accession and version. And then you would give us the C dot or G dot portion of that HGVS expression in the HGVS column. On the second row here is not a full example submission. I've provided some alternate ways to describe some of the fields in the second row. And for the variant, the alternative is to provide a description in genomic coordinates. So for example, for an SNV, you could give us the chromosome, the start and stop location, the ref, and the alt alleles. And one thing I will note is that you should pick one way or the other to describe your variant, so either HGVS or chromosome coordinates, but not both. So that's the first piece of information we need, is the variant. As we scroll to the right, we get to the second piece of information, which is the condition. And again, for condition, there's 2 ways that you can tell us the disease or the drug response that you are reporting the variant for. So our preference is that you give us a database identifier for the disease, so something like an OMIM number. So that's what I've shown here in the first example. To do that, you would fill in this column condition ID type. So that's the type of identifier you're giving us, the database that it's from, and we've got a list of the allowed databases here. And then condition ID value is the identifier itself. So this is MIM number 232300. The alternative is that you can give us the disease name, and we think this is appropriate when there's not an existing database record for the disease. We do want to leave the door open for discovery and new things. But if there is an existing record, using a database identifier is a more specific way to submit this data. And again, you should pick one of these options, but not both, either a database identifier or a name, but not both. So that's the second piece of information we need. The third piece of information is the interpretation. So that's the green section here. So the clinical significance is a term that we've also used for interpretation. So that's where you tell us how you classified the variant. The ACMG terms are here, as well as a few other options. And then you can see several of the optional fields that we talked about here. You can provide the date it was last evaluated. You can provide your assertion method or your assertion criteria. So this is the document that describes how you classify variants, and this is one of the requirements to get your star on your Clinvar submission. And there's 2 ways to do that. You can provide it as a citation, like a PubMed ID. This is common if you use, say, one of the published ACMG guidelines or published guidelines from an expert panel. In that case, you tell us the PubMed ID as the assertion method citation. And then as the assertion method, you give us a name for that method. And so this would be something that we could use on the website as the text for a link to link out to this document. The alternative is if you have a file that describes your rules instead. And in that case, the assertion method citation is the name of your file, and you'll provide that file to us in Submission Portal when you submit. And then again, the assertion method is the name that we could use to link to that file. Other optional pieces of information here that you can give us about the clinical significance are the mode of inheritance. You can give us citations, and then you can give us your comment on clinical significance. That's the blurb that you see on the website. And you'll notice here the example I pasted in ACMG codes that have been used for some variants in Clinvar. We don't have a structured way to provide any of the codes that are recommended in interpretation guidelines. So one thing that some submitters have chosen to do is include these codes that apply to their interpretation as part of this comment, and I think that's really appreciated by users. So the last piece of information is the evidence. And you remember for this type of record, the evidence is just a single observation at the aggregate level with not much information. Most of the detail was provided as part of the interpretation. So here we're just saying the collection method is that it was from clinical testing. The variant was seen in the germline. The affected status of individuals with the variant is unknown. And one thing I will note, we talked earlier about updating your records. If this were an update, these last two columns here are where you would indicate that. So in Clinvar accession, you tell us your SCV accession number for your record that you're updating. And then in the novel or update column, you explicitly let us know that this is an update. If this were a new record, you could just leave both of these columns blank and we would process that correctly. So that's our first example. So that's a simple submission for an SNV, just a single aggregate observation as the evidence. The second example we're going to look at is the CNV that just has a single case level observation as its evidence. So again, we're starting on the variant tab in the full spreadsheet, and you can see I've skipped over these first few columns. You can describe CNVs with HDVS, but our submitters typically do not. So I'm going to skip over here to the columns that focus on chromosome coordinates. So for a CNV, a typical way to describe that is to tell us the chromosome and the variant type. So here I've described a copy number loss. Other options are listed here. It could be copy number gain or other kinds of variants. And then you provide the coordinates. You can see I've hidden the columns here for absolute start and stop, and that's because typically CNVs are detected on microarrays where you don't know the precise location of the variant. You know a more imprecise location based on the location of the probes. And so inner and or outer start and stop are usually the more appropriate type of location. We've got more information in the instructions if you need a description of inner and outer starts and stops, but here I've represented the location as inner start and stop on chromosome 7. And then you can also tell us the copy number of the variant. So copy number is the observed copy number. This is a copy number loss. So we observed 1 copy, and the reference copy number, what you would expect to see, is 2. So that defines the CNV itself. The second piece of information is describing the condition. And this is a little bit more difficult for CNVs. So what we learned from our submitters is that typically for CNVs, there's not a named disease that you are interpreting the variant for. In some cases, there may be, and if that's the case, you can provide the condition just as you did on the last example. You can give us a database identifier or a name. But often there's really just a set of clinical features that were observed in the patient, but it's not necessarily that you are interpreting the variant for that entire set of features. And in Clinvar, we do distinguish between a disease that you're interpreting a variant for and clinical features that were observed in the patient. So this value here, condition, is really what disease did you interpret the variant for? And if there's not one, we have 2 options for you. So one is you can just tell us not provided. I am not providing a disease that I interpreted the variant for. That is okay, but I have to tell you, Clinvar users don't love that information. They really want more information about phenotype. So if it's possible for you to provide those observed clinical features, we do recommend that as a condition, as sort of a condition name, you use the words C cases. And it's not really a disease name, it's really more of an instruction to our users to say, see the case level information where there's phenotype information. So we don't want, in that case, we don't want people to think that there's no phenotype information at all. We just want to direct them to the case level information that is available And we'll look at those clinical features on this submission in a minute. So that's the way that we've worked CNV disease information into the Clinvar submission. The third piece of information is the interpretation. This is fairly similar to the last example. So you tell us the clinical significance, any of these optional fields that apply. And then here's your observation. As we looked at on the conceptual example, this has a little bit more information than you might provide for a SNV. You still provide the minimal fields of the collection method, the allele origin, and the infected status. For affected status here, I would say it's yes if you know observed clinical features in that patient. And for the clinical features, there's two ways to provide this information. You can give us the name of the feature or the HPO ID. Either way, we do ask that you use HPO names or IDs if they're available. We know that HPO is not comprehensive, so if you have a new feature that's not been described there yet, you can just provide the name to us. But we do ask that this is a list of clinical features rather than a prose description. And if you do want to provide that prose description of the features, you can provide that as a comment that would supplement the list of features. And then finally, there's this extra field here about the method to describe how the CNV was detected. And there's a list of the allowed values here. Okay, so that is our CNV example. And then the last one we're going to look at is the more complicated examples. This might be something that is more likely to come from a research group that has the consent to provide lots of case level information for a trio. And for this example, I'm just using the same SNV that we looked at in the first example. So this looks very similar. So this is the same variant that we looked at on the first spreadsheet. It's the same disease. I provided the same information for the interpretation. But you'll notice I didn't provide any information about the evidence, the observations of the variant on this first tab. And that's because we're going to provide that case level information on the second tab, which is called case data. And because we need to link together information on two different tabs, in this case, we do need this identifier called the linking ID. So this ID can be any ID that is unique to this variant and this condition from your organization as the submitter in Clinvar. If this is an update and you've got an SCV accession number, that's a great thing to use as a linking ID. If you don't, you can make something up. So for example, here I've used the combination of the CDOT for the variant and the disease identifier. So you would take that linking ID and you enter that into each row on the case data tab where you're going to provide an observation of the variant. So this is a trio, so I've got 3 rows. So for this one, the minimal fields here are these 3. So I'm saying this submission is from research. The variant was observed in the germline in all 3 individuals. And here you can see the affected status. I've indicated that the first individual was affected. So this is the proband, and I've said no for both of the parents. I filled in clinical features for my proband. I described the date that the phenotype here was evaluated. And then you can see for each person, the proband and each of the parents, I filled in more specific information, the sex, age, whether or not they were the proband, and their zygosity for that variant. So you can see there's different ways to provide data depending on how much information you have, what level of consent you have, and how much time and effort you can put into structuring this kind of data for your Clinvar submission. I'll also point out there's a lot more information online about filling out different kinds of submissions and other kinds of data on the spreadsheet. And a lot of that information also applies to the wizard submissions as well as the API as well. And before we move on to questions, I just want to say a big thank you to the Clinvar team. On the left here is our team of developers, followed by our curators, our leadership within the medical genetics and human variation program for Clinvar, and our leadership within NCBI and the NLM. We have a few minutes for questions here now, but if there's anything that doesn't get answered today, you can always email me directly or the team, and we're happy to take questions at any time. So with that, I'm going to get my eyes back on the WebEx, on the Zoom here, and see if there are any questions.
Nothing has come in yet, Melissa, but oh, here we go. Can you speak a bit on submitter status 1-star, 2-star?
Yes. So the stars really represent what we call review status. And so this is to give you an idea of what level of review went into this interpretation. And this applies both at the level of a submission and at an aggregate record, so a whole variant record that you see in Clinvar. So for submitted records, the requirements to get from, say, 0 star to 1 star are that you have provided your rules for classification. So that's the assertion criteria that we looked at on one of those submissions. So that's a document that describes these are the categories that I use to classify variants and the criteria I need to put a variant into each of those categories. And then you also need to provide some form of evidence, and evidence here doesn't necessarily mean the minimal evidence. We have a list on the website. You must provide something like that comment on clinical significance, some citations, something more structured that really gives an idea of what information did you use to classify this variant. The expert panel submissions get the 3-star review status, and then we have a few records that have the 4-star, which is from a practice guideline. And 2 stars is specific to variant level records, a submitter, because it needs consensus among submitters. So individual records do not get the 2-star designation.
And I realize I missed one question. Lana asked, could you clarify the approximate processing time for submissions? Yeah, great question. So right now, I would say we typically get back to submitters with initial questions, definitely within the week and usually within a couple of days. After that, it can depend on how soon we can get any errors or clarifications resolved with the submitter. Occasionally now we have some issues on our end where it might take a little bit more time to get something processed. We don't have a guaranteed time because these things can vary from submission to submission. Our turnaround time right now is roughly 2 weeks, I'll say, average. Great, thanks. Any other questions from the audience? All right. Well, our session was supposed to end at 5:10, so we are 1 minute or less away from that. So we did well on timing. Looks like everybody's questions have been answered. So right now we have a break for just about 5 minutes to let you gather yourselves, and then we're going to be starting at 5:15 on Spatial Chat, and Sharon posted the link into the chat. That will include announcement of the awards. That will be a fun session, so don't miss that. We'll also hear the rapid fire poster 1-minute talks, and then much of the time will be spent with happy hour networking. And I can't speak highly enough about Spatial Chat. It's a really fun environment to move around and talk with people fairly close to what it's like in a real actual room. So encourage you all to come join us on Spatial Chat and hear the next activities, and then we'll see you tomorrow morning for the second day of the retreat. Anything else I missed, Sharon?
No, you covered it. And again, please join Spatial Chat. Heidi and I've had a lot of fun with it at other meetings.
Yes, my lab has
Remember,
her own submission.
tomorrow, if you don't, remember that tomorrow morning there's no central talk. So just pick the breakout you want and go directly there.
Yep, exactly. Thanks. All right, we'll see you all in Spatial Chat.